Good morning, ladies and gentlemen, and thank you for welcoming me here. And my name is Garmaa Gantsetseg, and I'm a second-year PhD student at the Institute of Translational Medicine Department. And my project thesis title is "The Role of Micronus in Renal Fibrosis." And so today I'm gonna talk about microRNA kidney disease today mainly. So my supervisor is Gabor Kokeni, and you can see also our team here at the Center of Translational Medicine Department. And as a young medical doctor and scientist, my vision is to contribute to building a healthy community which is supported by basic and translational medicine. So in terms of my doctoral study, I would like to investigate the novel microRNAs that play an important role in kidney disease. And so, in terms of this goal and my interest, I set the two specific goals with the program. The first one is to conduct a meta-analysis on microRNA for fighting studies. That means we are not gonna interest in few microRNA, we're gonna interest in tons of microRNAs that is tested in kidney disease. And second one is we are more interested in to determine the diagnostic accuracy of microRNAs that's already kind of proven in clinical settings. So first, I would like to introduce my first project, and the title is Meta-analyses of microRNA expression for fighting studies in different types of kidney disease and samples. You can also see our team here. And before I jump into results and method, I would like to introduce a little bit background here, why we have to study the microRNA in kidney disease. And 10% of world population is affected by this kidney disease, and those people can lose up to 90% of kidney function without experiencing any clinical symptoms. And so we have to take care of this disease actually. And so why microRNA? Because in the last 10 years, microRNA got an attention from scientists, and it is considered as a diagnostic marker for any kind of disease. It has the potential of inhibiting target genes, and it also affects the protein expression. If you look at this scheme, microRNA have a lot of interaction in human body, also in any kind of animal. And so it can also have activators, effacers, and targeted genes, and it can also represent the pathways, and it also can interact with the drugs too. So in terms of this theory, we set a specific goal. What are the most dysregulated microRNAs in human and animal CKD? We are interested in human and animal. We set the PICO, and P will be humans and animal tested for microRNA for fighting studies. That means tons of microRNA, and exposure will be the presence of kidney disease, and comparator will be totally healthy animal, and human outcomes will be most dysregulated microRNAs by ranking order. And we set this specific goal, and by the hypothesis, we believe that maybe we can diagnose the kidney disease using this microRNA. And since we are using a lot of analyses, I would like to introduce the methodology of my project here. First, microRNA is registered like almost 20 years ago, and there's a lot of studies that there's a big database, and so we're interested in any type of samples, and the microRNAs has to be tested with array or sequencing, any other techniques. And so we did a systematic search around like 17 articles, and finally we ended up human 32 and animal 13 studies. After the data extraction, we have to do the robust aggregation. That is the main analysis of our project, and we put the all microRNA list in this way, and we ranked all microRNAs in Excel. After that, we did the analysis on our program. And then we we find out the target genes, and after that, we did the enrichment analysis using bioinformatic tools. And so now I would like to go for the results, and results will be mostly from robust aggregation. Outcome will be most up or down regulated microRNAs, and subgroups will be human and animal, also the different kidney disease by etiology, and third, the different sample types, urine, blood, or tissue. And after data extraction, we set the database like human studies 1 to 6-fold, for animal studies 1 to 2-fold. And so today I'm going to introduce these two results to you. These slides will show you the results from robust rank aggregation in here, it's from the mouse kidney tissue, and you can see the first 6 microRNAs are most upregulated in animal kidney model. And the second slide, I was interested in human sides, and it's from urine sample, and it's most upregulated microRNAs, but only 1 microRNA was significantly upregulated. But also in urine, only 1 microRNA was downregulated. And so further on, I was interested what's happening there, how we can connect with this microRNAs and what's going on in the human body. And so we pick up the urine samples, and with the enrichment, pathway enrichment analysis here, if you look at these graphs, the bigger dots will say these pathways are more significantly enriched in kidney disease in urine sample, and we found out the MARC signaling pathway and cellular senescence pathway was more significantly enriched in kidney disease in urine sample. And after that, we also enriched the biological process of these 2 microRNAs, and so we found out this biological process in human kidney disease. And so here, these slides show you the summarize the results from microRNAs. And if you look at these tables, there is a different type of sample types and microRNAs. It also shows you the direction of up or down regulation. And so in here, I also have to mention, we look at the animal data. We believe that we are doing a lot of data for animal, but we really cannot translate these animal results to human sites. And so we also have to consider in the future. And so after that, we also have an interest to build the network analysis based on this lot of microRNAs, and we will see the bigger picture and soon. So now I would like to introduce the study implication. So for the clinical side, we believe that maybe we can bring up the most important microRNAs that could be tested in clinical settings in kidney disease. And in a translation side, maybe we can find out the new molecular pathways that play an important role in kidney disease. And it's worth to mention we also had the strength and limit. It was really hard to pick up the raw data because there was a lot of lack of data. And we only included the mouse studies because of the heterogeneity of studies. And there was a different type of sample types too. So my second project is the diagnostic accuracy of microRNAs in chronic kidney disease. In here, the evidence level will be much higher, and you can also see our team here. And a little bit about background. Nowadays, we are using serum creatinine and EGFR and proteinuria as a diagnostic marker of kidney disease, but these are not enough for us to diagnose diseases, especially for the early stage. And so, as I told you before, microRNA is kind of diagnostic and therapeutic markers. And so there is some also evidence, some microRNAs that play in high sensitivity and specificity. And if you look at the left side of the screen, kidney biopsy is actually the gold standard of kidney disease diagnostic, but it's kind of risky. In that case, we have to find an invasive method to diagnose disease. And we asked the question, what is the diagnostic accuracy of these microRNAs? And we set the PICO framework, and it's better to also mention clinical implication of this study. There is a lot of studies, but we do not know what is the diagnostic accuracy of this biomarker, and we need to find out this one. And we did the systematic search, following numbers we found out, and now we are working in the title and abstract selection. It's an ongoing project now. So you can see the total framework here, and the project deadline too. And so thank you for attention. I would like to mention Mongolian proverb. It means where there is a will, there is always a way. Thank you very much.
Thank you, JJ, and congratulations. And thank you for showing us this new dimension of meta-analysis, actually. I wanted to ask, why do you think there is a difference between the profiles of microRNA dysregulation between human and animal? Do you think the animal models are imperfect? Should we consider other models? Or why do you think there are these big differences?
Thank you for the question. In everyday life, as a basic scientist, we are working in mice or rat, and we got a result, and we believe that that's going to translate into human side. And if you find out, like, for example, microRNA 1, and we believe that it also going to apply for human side. And so we buy a lot of stuffs and try to implement in this human side. But I think there has to be difference, because these are the totally different biological organism. And if you look at the molecular pathways, it's also different. And so if you are interested in clinical implication of any kind of studies, it's better to also try to check the clinical data too. And so that's why we are interested in human and animal, both sides of the history.
Thank you very much for your presentation. It was fantastic. My question is about, to your knowledge, has there been any clinical studies which utilize this early diagnosis screening by microRNAs and then an intention to treat? Because the pre-assumption that we have is that early diagnosis leads to better outcomes. But with this modality, are we sure that early diagnosis and screening is going to lead to better outcomes? Thanks.
Thank you. It's a very fantastic question. And so there is only one ongoing clinical trials. They were focused on microRNA 21 in kidney disease. But it's really early to say what's going to be the results from these clinical trials, but it's ongoing. We don't have any data. So it's conducting in America, and so it's too early to say. Yeah.
That can be your results of the first, I mean, those many, this one, the next one. So based on your results, actually, can you suggest that any of these markers, I mean, any of these could be used as biomarker? I mean, is there any of these, I mean, just let's concentrate on the human, so, I mean, on the line of diagnosis. So, what do you think? Or more studies are needed, or what?
Thank you very much. It's very important to translate the basic science to clinical science, and so we also think about this question during this project. Using this up or down regulated microRNAs, we can make the diagnostic panel. It means the diagnostic small chips, it's already applied in prostate cancer using microRNAs. They put these 5 to 6 microRNAs in one chip, and they drop the blood or samples, and they will see how it's up or down regulated. And so if it shows some result, it means that they can diagnose prostate cancer. Maybe in the future we can also use these results to see in the kidney patients.